Hi everyone, Ryan Balut here, neuroscience undergraduate student at the University of Toronto, and I'm with you today to talk a little bit about machine learning implications for protein structure prediction. So without further ado, let's get into it. You can think of proteins as activity molecules, executing a number of functions that are imperative to cell survival. In biochemistry, we learned that the structure of a protein determines its function. If you'll notice on the left of the screen, this is creatine kinase, a protein enzyme involved in cellular metabolism and the movement of muscles. Now, in order to appreciate the combinatorial nature of this protein, you'll need to understand the 4 levels of organization and what exactly goes into determining structure. On the 4 organizational levels of proteins, we have the primary structure, also known as the amino acid sequence. We have secondary structures that are universal to all proteins. There are tertiary domains that make up quaternary structures that ultimately form functional proteins like the ones seen prior. Machine learning applications require primary amino acid sequences. It's with these primary structures that algorithms are able to predict tertiary structure. Let's take a look at how accurate these predictions are being made. Highlighted in green is an experimentally derived structure from the researchers at Nature, Jumper, Evans, among others. In blue is an algorithmically derived structure from a machine learning platform known as AlphaFold. Before revealing the accuracy of this program, I'd like to direct your attention to the figure in the top right and notice the specific arrangement of amino acids around a metal ion. This protein cofactor is involved in recruiting positive or negatively charged amino acids in their specific orientation to help with protein folding. If you'll notice, the experimental and predicted amino acids are almost in the exact same conformation. The middle image shows a protein domain, a tertiary structure, with alpha helices and beta sheets in seemingly identical positions. The bottom structure helps us to realize the 92% accuracy of AlphaFold. This incredible achievement of AlphaFold can be attributed to the advancements in machine learning technology and also the accessibility and amount of protein data available on the PDB. As of 2024, there are 100,000 unique protein sequences correlated to their experimentally derived protein structures. Now, the potential here is for machine learning programs and researchers alike to relate and devise pattern recognition processes to associate individual amino acid codes to their experimentally derived structures. This figure shows the entire process of protein structure prediction, but I'd like to pay specific attention to the inputted amino acid sequence on the far left and the reference to the databank on the top left. Now, programs have access to PDB data and can reference inputted sequences with others in a process known as multiple sequence alignment. Directing your attention to the left of the screen, notice the ladder arrangement of similar sequences. More on multiple sequence alignment, homologous sequences are overlaid to determine the strength or likelihood of specific residue contacts. This information is extracted in both a co-evolved manner as well as with attention to specific amino acid preference. Keeping in mind our conversation on sequence alignment, I'd like to take us through the convolutionary neural network located at the bottom of the screen. The statistical inferences made through this rigorous process are outlined in the top right by the letters A through F. If you'll notice, parallel Gaussian distribution models correlate with beta strands and beta sheets, while perpendicular and evenly spaced distribution models correlate well with helices. This type of explainable AI and deep learning serves as a unique reminder for the potential of ML in biochemistry. In conclusion, laborious methods of experimental structure determination that used to stand in the way of discovery seem soon to be replaced by machine intelligence. Let's let biochemists determine sequence, use algorithms to predict structure, enable scientists to interpret function, and allow visionaries to make discoveries. Thank you so much for listening to this talk. Here are my sources. On behalf of myself and the U of T community, take care.